Hello, everybody. We're going to move on to Chapter 4. And again, with Chapter 4, you want to read through your study guide, guidelines, and so forth very closely because I'm not requiring this entire chapter. There are quite a few things that we're going to leave out. Kind of to get started, all right, so eukaryotic cells, prokaryotic cells, we've been talking about that pretty extensively. And eukaryotic cells in general are larger and more complex. They have a lot more internal structures that have their own membranes. Those are special compartments that carry out specialized functions. So at the first part of Chapter 4, hopefully you're keeping up with your reading, but there is some discussion about the theory of endosymbiosis. So a symbiotic relationship is where two living things get together and become dependent upon each other in some way. And so there's a lot of speculation about where did these first eukaryotic cells come from? How did they develop? And so there is a theory called the theory of endosymbiosis, where there was some sort of ancestral cell, not quite a eukaryotic cell yet. And some people believe that this was an archaean. That's kind of some of the current thinking about this. And then the membrane may have been more flexible than we typically find today. And at some point, we had a pre-eukaryotic cell of some type that engulfed or took in a prokaryote, probably bacteria. And then they developed a symbiotic relationship with each other. So where you've got the bacteria living inside this other type of ancestral cell. And the thought is, why was that advantageous? Probably because the little bacterial cell that was engulfed was capable of using oxygen very, very efficiently. We'll be getting into that more as we move into chapters 6 and 7. It's an aerobic type of organism that had developed the ability to use oxygen gas to its advantage to help it make lots and lots and lots of ATP fuel. So you can imagine if this little pre-eukaryotic cell over here didn't have that capability, and it took in this bacterial cell that did have that capability, now that the little engulfed cell is making ATP that bigger pre-eukaryotic cell could also use. And the little cell has protection from the bigger cell by living inside in there. And so over time, it's thought that's where the mitochondria came from. Remember, the mitochondria are the powerhouses of eukaryotic cells. That's where where ATP is largely produced, oxygen gas is used. And so over many, many, many generations over time, those bacteria lost a lot of their independent characteristics, and really they just became nothing more than little machines inside cells that crank out lots and lots and lots of ATP. So there's a lot of evidence that this is likely what happened, because mitochondria in eukaryotic cells like we have actually have some of their own DNA. And if you look at that DNA at a detailed level, it has a lot of similarity to the DNA of some bacterial species that still exist on our planet today. So keep in mind, all this was happening billions of years ago. And also, mitochondria divide, and new ones get created when a cell needs new ones. They kind of have a bacillus shape, so there's some evidence there for mitochondria having originally been independent bacteria that at some point set up a symbiotic relationship with another type of ancestral cell, and over time the two got together and created this larger, more efficient type of cell that has become known as a eukaryote. And also over time, these other compartments developed inside these eukaryotic cells as well. And over many, 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 many generations, they ultimately developed into the more complex types of eukaryotic organisms we have today. Also, your plants and your algae, the ones that do photosynthesis, kind of a similar theory exists, endosymbiotic theory, where you had eukaryotic cells that engulfed photosynthetic bacteria that you see there. Those photosynthetic bacteria were capable of harnessing energy from sunlight to make ATP. And they also had the capability of manufacturing their own complex organic molecules, starting with carbon dioxide. So they took in carbon dioxide from the atmosphere as well. And so plants and algae likely developed from those types of bacteria setting up an endosymbiotic relationship and bringing this capability of doing photosynthesis to some sort of ancestral eukaryotic cell as well. Let's call the theory of endosymbiosis. And remember, a scientific theory is something for which there is a lot of scientific evidence to back it up. It's not something that's 100% settled and is not still undergoing changes and modification, but a scientific theory is pretty well substantiated, or generally very well substantiated. All right, so this is a picture from your textbook of a generic eukaryotic cell, and I notice down here they have the generic bacterial cell, and again, kind of what they're trying to emphasize is the size difference, and it's even more dramatic than that. Typical bacteria would probably be about like that next to a eukaryotic cell, but there is a lot of diversity with eukaryotic cells. They're not all alike. And all right, so in your textbook, it goes through all these different internal parts of eukaryotic cells, such as the Golgi apparatus and the endoplasmic reticulum and so forth and so on. And you've probably learned about those internal structures. We were just talking about the mitochondria. There's one there. And you may have discussed those internal structures of eukaryotic cells in a freshman or high school biology class, or in your ANP1 class if you've already taken ANP1. Hopefully you have, but not everybody has. Some people take micro first. And so what we're going to do, I'm not going to go back through all of that information. I'm not going to test you on it from Chapter 4. But keep this in mind that this information is available in Chapter 4 as a reference, and then later in the semester, as some of these more complex cell parts come up in discussion, we will return to or discuss more specifically what we need to know about those cell parts at that time. All right, and your textbook has a pretty good little flow chart, kind of like they did for the bacterial cell, eukaryotic cell, and then some of the various structures that can exist. I don't know why it says external structures up at the top. That's an error. That should say eukaryotic structures and not external structures. But again, just kind of familiarize yourself with the names, and I'm not going to test you on the details of what those do. We'll just return to those during the course when we need to know something about a particular eukaryotic cell structure and what it does. Now, a lot of things are similar, but kind of just in some key things, eukaryotes versus prokaryotes. Prokaryotes generally have more going on at the cell surface, cell wall, those different layers that are there. Cell membrane bacteria and eukaryotes generally functions in a pretty similar way. The inside of a eukaryotic cell has more of those membrane-covered compartments like endoplasmic reticulum and Golgi and mitochondria and so forth, and you don't find those in bacteria. The nucleus, eukaryotic cells have a membrane-covered nucleus, bacteria do not. And your eukaryotic cells, again, are generally more complex and larger, more organized internally. You have more of those membrane-covered organelles. You don't have those in bacteria. Okay, just as a review, what microorganisms fall into the eukaryote category? So we got 4 major groups that we want to discuss. Those include the fungi, protozoans, helminths, and algae. And you're getting some of this on your Learn Smart labs as well as you work through that. So what we'll do in the next video lecture for Chapter 4 is we'll talk a little bit more about fungi. And so please read through the information in your textbook if you have not done so already, and I'm going to hit on some of the highlights and bring in a few examples I want you to know about, a few things more specifically about those types of fungi. And we'll do the same for those other eukaryotic groups as well.